Enzymes are nature's catalysts. They're extremely efficient. And if we could harness the power that nature has generated for things that are part of our world, of our man-made world, then we could replace industrial processes with new enzymes. I'm Florian Hohlfelder, and the purpose of my group is to understand how proteins work and make them useful for applications in chemical biology and synthetic biology. Enzymes are the most efficient catalysts that are around, but we don't know how to make them from scratch. So we'd like to know where we can find useful or interesting enzymes in the vastness of sequence space. Proteins are built from amino acids, and there are multiple permutations, actually too many to ever screen. And so we need rules or technologies, or at least a strategy to explore this enormous theoretical diversity that's there.
My name's Lorna Richardson, and I look after the day-to-day running of the MagnifAI resource. So MagnifAI is EMBL-EBI's microbiome data and analysis resource. A microbiome is the combined genetic material of all the organisms that are contained within a particular sample, for example, soil or marine water or human gut, things like that. Florian's group were interested to mine our protein sequences that we hold, looking for specific enzyme sequences, and we were able to help them do that by using some of the metadata to help them improve their searches.
And the MagnifAI database helps us then to take sort of one hit that we get experimentally and find hundreds of enzymes that are similar. They have similar function, but often have properties that are better. They're more stable, they're more evolvable.
What's interesting is these sequences are very often not represented in the reference databases, so they really represent enormous novelty in terms of nature's bioengineering.
The combination of microfluidics, metagenomics, and MagnifAI give me access to a huge collection of enzymes that I can explore here in the lab. With our technology, we can screen millions of droplets per day, and each droplet contains a different enzyme. And our task is to find the most interesting enzymes from all these droplets. So thanks to the sequence of this candidate, we can use MagnifAI to explore even more. So literally magnify even more what we found in the lab.
So MagnifAI is probably the most comprehensive database, and it's easy to use, it's well curated, and it keeps on growing.
MagnifAI is just one of many open data resources available at EMBL-EBI. So we're interested in genomic data, but the other resources, they cover a whole range of data types from genomic, protein, chemical, and many others. EMBL-EBI's resources are really crucial to researchers who are addressing today's major global challenges, things like disease prevention and food security and loss of biodiversity.
There are lots of things where biocatalysis could be useful, by making food products, by making new therapeutics, by degrading plastic in the environment, by bioremediation, by detoxifying nerve gases, pesticides, and bioterrorism agents, for example. If we do that, we could do everything that now works in factories with green reagents, with enzymes that don't have toxicity, are incredibly efficient, and replace the current economy with a green economy.